So in this Insta lecture session, our focus would be on Burkitt lymphoma. We try to understand why Burkitt lymphoma is the fastest growing human tumor. What are the three clinical variants of Burkitt lymphoma, namely the endemic variant or African variant, the sporadic variant, and the immunodeficiency associated variant. And we try to understand how they differ from each other. We try also to focus on the how do you diagnose a case of Burkitt lymphoma, what are the markers which help to diagnose a case of Burkitt lymphoma. Here I also like to mention that nowadays all leukemias and lymphomas, they are diagnosed with the help of flow cytometry. And in flow cytometry, what we do, we try to locate specific antigens which are expressed by the leukemia on the lymphoma cells over their surface. So each leukemia and lymphoma, they express a set of specific markers over their surface. Often they are CD markers, cluster of differentiation markers, and these markers, the set of markers, actually like an identification ID for them. Each one expresses a specific set of markers. Like previously in CLL or /SLL, I told you they express CD5, CD19, and CD23, all of them, but they do not express other markers like T cell markers like CD3. So we'll be also focusing on this thing. Now, before I go into discuss about the Burkitt lymphoma, I need to tell you a very important story dealing with the discovery of Burkitt lymphoma. Now, Burkitt lymphoma carries the legacy of a famous Irish surgeon who contributed enormously, not only for the discovery of the Burkitt lymphoma, but also he has role in understanding the etiopathogenesis of colorectal cancer. Now, he was Dr. Dennis Burkitt was a Irish surgeon who was working for British military group during the World War II, which happened between 1939 to 1945. And during that time, as he was serving the British Army in the Equatorial Africa, or in the sub-Saharan African region, he was quite moved seeing the poor health status of the African people. So when the World War II ended in 1945, he decided to spend the rest of his life serving the Equatorial African people, and he moved to Uganda, in a country in the sub-Saharan Africa called Uganda, and he settled there in Kampala. And in his clinic, he used to see a lot of young kids. They're coming with a jaw mass, in their facial region, and very frequently he used to observe that these kids used to die very soon after visiting him. They never used to come for the follow-up. And often they have cancer in other sites also, not only in the jaw, they have cancer in the stomach, ovary, kidney, and other sites also. So he became curious and he tried to document all these cases. And in 1958, he published that he demonstrated that this group of cancer is basically a unique kind of pediatric cancer. And later, this kind of cancer was known as, got its name, Burkitt lymphoma. That's the basically idea how Burkitt lymphoma was discovered. And Burkitt lymphoma is also associated with the discovery of the Epstein-Barr virus, but I have tell that story in a different Insta lecture. Now let's focus on Burkitt lymphoma again. So Burkitt lymphoma is the fastest growing human tumor, and it has it's a highly aggressive variant of non-Hodgkin lymphoma. And like non-Hodgkin lymphoma, they used to prefer extranodal involvement. That means non-Hodgkin lymphoma, unlike Hodgkin lymphoma, has a tendency that they do not always involve lymph node. They involve sites other than lymph node, extranodal involvement. And Burkitt lymphoma has three unique subtypes. One is known as endemic variant, also known as African variant. Second subtype is known as sporadic variant. And the third subtype is known as immunodeficiency associated variant. Now the first type, which was basically described by Dr. Dennis Burkitt, is known as endemic variant or the African variant. And this type, they typically present in a young kid, 4 to 10 years of children, particularly more common in the boys rather than girls. They present with a jaw mass or the facial mass involving the maxilla. Now they can also involve this kind of cancer can also involve other sites also, like as I mentioned, breast. They can involve the kidney or the adrenal, and they are seen in the regions where there is malaria is endemic, and they show very strong association with Epstein-Barr virus. Almost 100% of them, they are associated with Epstein-Barr virus. This is all you need to know about the endemic or the African variant of the Burkitt lymphoma. The second variant of Burkitt lymphoma is known as sporadic variant. This is more common in Northern America and the western part of the Europe, and they typically present with abdominal mass with ascites. This kind of Burkitt lymphoma often present with a ileocecal mass, which is often detected by the CT scan or other radiologic methods. And jaw mass or the facial mass involvement is much less common. And this variant also shows a less stronger association with the Epstein-Barr virus. The third variant is known as immunodeficiency associated variant, also known as HIV associated variant, and they often present with generalized lymphadenopathy with other features of immunodeficiency. And Burkitt lymphoma is one of the common lymphoma which shows a very strong association with HIV. Now, coming to the other aspects, pathogenesis, Burkitt lymphoma shows a characteristic overexpression of the MYC gene, which is located on the long arm of the chromosome 8. And they are associated with certain characteristic translocation, like translocation 814, which is seen in almost 80 to 85% cases, and the rest of the cases they can show also translocation 28 or translocation 822. And the common feature of all this translocation is this, that the MYC gene, which is located on the long arm of the chromosome, as a result of the translocation, they sit next to some immunoglobulin associated genes, either on the chromosome 14, 8, chromosome 14, 2, or other chromosomes where these immunoglobulin associated genes are there, or 22, or other these genes are there. And all of them, they result in overexpression of the MYC gene. And the overexpression of the MYC gene are associated with excessive proliferation and also plays a crucial role in the metabolism of the cancer, because MYC gene overexpression also leads to overactivation of the Warburg effect. Warburg effect means that unlike the normal cells, the cancer cells usually use the aerobic glycolysis method. Normally, we normal cells usually use the glycolysis method only when there is no oxygen is available. But the cancer cells, even in the presence of oxygen, they use glycolysis method because that helps them to proliferate rapidly. It has been found. This is called Warburg effect, as was described by Dr. Warburg, who got Nobel for this. Now, coming to the diagnosis, obviously they are diagnosed by the biopsies, and in the biopsies, the histology, what we see two characteristic features that describe. One is called moth-eaten appearance, another is called the starry sky appearance. And moth-eaten appearance is usually seen due to focal areas of coagulative necrosis or hemorrhage. And more popular term which we use for this is called starry sky appearance. And starry sky appearance basically is seen because the tumor cells have a very high apoptotic rate and high proliferation rate. So often those apoptotic tumor bodies are eaten or engulfed by a kind of macrophages known as tingible body macrophages. And these tingible body macrophages have a very large pale cytoplasm, and they create an appearance like the stars among the basophilic tumor cells, which are the sky. So this creates an appearance which creates an appearance like the starry sky appearance. And the starry sky appearance, the idea is thought to be inspired by the famous Dutch painter Van Gogh, because Van Gogh has drawn a very famous painting known as Starry Sky, and I think that inspired some pathologists to think this appearance as a starry sky appearance. And flow cytometry or the immunophenotyping, they express certain characteristic markers. Those markers are CD19, CD20, and CD22 because they are basically mature B cell neoplasm. And as it's also a germinal center tumor, they also express CD10 and BCL6. So these are and obviously in translocation they also present characteristic translocation 814, which I already mentioned. The other things I've discussed in the coming lecture. Thank you.